DNA, when it was first described, was described as a right-handed spiral, which was called as aDNA. Later on it was discovered that this aDNA, which is a right-handed spiral, is in a dehydrated state. With hydration, it opened up slightly more, but it remained right-handed spiral, which is called as a bDNA. So bDNA is a state in which the DNA is in a living cell. If the DNA gets methylated, it reverses the spiral and becomes a left-handed spiral, which is called as zDNA. For the mRNA and protein synthesis, the spiral DNA has been despiralized. zDNA, when it's trying to be despiralized like the bDNA, actually gets hyperspiralized. So it remains condensed, does not produce proteins. And that inactivated part of the DNA which do not produce proteins is called as heterochromatin. Those which are producing proteins are called the good chromatin or euchromatin. Cells which produce many different types of proteins in large quantities have a greater part of the nucleus in euchromatin phase, and those which are largely inactive cells have more heterochromatin. Nuclear swap experiments of a hepatocyte nucleus exchanging with a fibroblastic nucleus. The fibroblast nucleus, which was largely inactive, when it enters the hepatocyte cytoplasm, becomes active. And the highly active hepatocytic nucleus, when it is transferred to a fibroblast cytoplasm, becomes inactive. In other words, the activity of the nucleus and the inactivity of the nucleus is decided by the cytoplasm. That's why cytoplasm is the boss to the nucleus. Another experiment which was done was between a cancer cell which keeps on dividing with a neuron which never divides. When the nuclear swap experiment took place between these two cells, the nucleus of the neuron, when it entered the cancerous cell, continued to divide. And the cancerous nucleus, when it was put in a neuron which otherwise always divided, stopped dividing, implying that even cancerousness lies in cytoplasm. The position of nucleus is like a henpecked husband who says, I am the boss of the house and I have my wife's permission to say so. Okay? Then we discussed about gender. Let us re-summarize the gender. Y chromosome and X chromosome, their P-terminal end are homologous. So that portion of the X and Y chromosome are called as pseudo-autosomal region because they are homologous, like autosomes. That's why X and Y chromosome pair up with each other during meiosis end to end, not side to side. Because non-homologous portions will not pair up. Only homologous portions will pair up. Close to the pseudo-autosomal region on Y chromosome, there is an SRY gene, sex-determining region of Y chromosome, which produces a protein called as TDF, testis-determining factor. During crossing over of X and Y chromosome, there's a potential chance of that SRY gene from Y chromosome transferring itself to the X chromosome. Individuals which possess SRY gene are said to be genetically male. Individuals who have one X and one Y chromosome are called as chromosomally male. Individuals who have testes are called as gonadally male. Individuals who have internal and external genitals like male are called as genitally male. At the age of 2 to 3, when the child behaves like a male, picking up toys which are noisy, are psychosexually male, production of excess testosterone after puberty, the individual is said to be hormonally male. SRY gene, although determines testis, 2X chromosome determine ovary. So if the individual has got 2X chromosomes with the SRY gene, will produce testicular proliferation of the medulla of the gonad and ovarian proliferation of the cortex of the gonad. This gonad will be called as ovotestis. Presence of ovotestis in an individual or separate ovarian and testicular tissue in the individual, that individual is called as true hermaphrodite, named after Greek god Hermes, who married Aphrodite and a child called Hermaphrodite. If an individual has got only one gonad, that is the true gender of the individual. If the gonad and genitals do not match, the individual is called as pseudo-hermaphrodite. Testicular tissue with female external genitals is called as male pseudo-hermaphrodite. Ovarian tissue with external genitals male-like or ambiguous is called as female pseudo-hermaphrodite. 3 common causes of pseudo-hermaphroditism in males. Testosterone converts the external genital to become male-like, but more potent than testosterone is dihydrotestosterone. Testosterone is converted to dihydrotestosterone by an enzyme called 5-alpha reductase, which is an autosome which is produced by autosomal genes. 5-alpha reductase converts testosterone to DHT, dihydrotestosterone, which is more potent in converting the external genitals to become male-like. Deficiency of 5-alpha reductase, an autosomal recessive condition, produces less amount of DHT, therefore external genital will become ambiguous. Testosterone is present, but external genitals do not properly form. DHT shows receptors in the external genital region, which is the protein produced by X chromosome. If that protein is defective, the receptors are defective, DHT cannot activate the external genital region. Again, the external genital region will become female-like. Here, the receptors are faulty, that means the external genital will not form male-like at all. This condition is called as testicular feminization syndrome, male pseudohermaphrodite. Testosterone is also produced by adrenal cortex zona reticularis, sex steroids. And this is produced in females as well. An androgen secreting tumor of the cortex of the female fetus will cause the external genital to become male-like or ambiguous. The androgen secreting tumor is the cause of female pseudohermaphroditism. Then we went into discussing the DNA. We just described that the dehydrated DNA which was first discussed was called aDNA, the hydrated state is bDNA, and the reverse spiral is zDNA. The zDNA is a condensed form of DNA, which is also called as heterochromatin. In female somatic cells with two X chromosomes, one of the X gets inactivated from a center present in the Q arm. The center is called as X inactivation center, which produces the protein called as XIST, X inactivation specific transcript. This XIST not only methylates the same X chromosome, it also suppresses the other X chromosome not to produce XIST. So that one X becomes inactive, one X becomes active. The inactive X also has some portions which are active. And both these X chromosomes, active as well as the active portion of the inactive, together are responsible for production of ovaries. Which is why Turner syndrome, classical Turner syndrome with 45X do not produce proper ovaries, and 47XXY do not produce normal testes, because the two X chromosomes which suppress the formation of testes despite having Y chromosome. Testes will be produced but probably non-functional. The inactive X chromosome in the female somatic cells started occurring 15th day after intrauterine life. On the 15th day after intrauterine life, there are around 5,000 cells. In female somatic cells, all these 5,000 have got two functional X chromosomes. On that day, inactivation of X occurs. And randomly, half of the X getting inactivated are paternal, half of the X getting inactivated are maternal, purely at random. After that, clonal generations of one particular cell will produce the same X which is inactivated in the parent cell. For example, in the 5,000 cell, let's take cell number 1. Here the maternal is inactivated. Say, when it divides into two cells, both the daughter cells will have the same paternal getting inactivated generation after generation. Okay? That's one point. This inactivation can be seen under light microscopy as a dark rounded body in the nucleus of female somatic cells. This was first described in the 1960s by two scientists, Barr and Bertram. And this inactivated one is called as Barr body. Somewhere 5 to 10 years later, in 1971, Mary Lyon LYON, stated that this bar body is because of inactivation of one X chromosome, and that this inactivation is random. This was called as Lyon's hypothesis. The enzymatic levels produced by X chromosome in males and females are equal, because the inactive one does not produce the enzyme. Active one is only one, and males also have one X chromosome without any bar body. In females, if they have a defective gene, say of factor VIII, hemophilic factor, one X is hemophilic factor, other is normal. One derived from one parent, other derived from the other parent. 50% of the X hemophilic gene, that X will become inactive. 50% of the normal genes will get inactive. But 50% of the chromosomes, cells which have got normal X which are inactive, which are active, will continue to produce factor VIII. And the production of factor VIII will depend on the need of the body. That's why despite carrying a defective gene, the factor VIII production in males and females continue to remain the same. That's why this condition is called as a recessive condition. One X is sufficient to produce whatever is the factor VIII required. Half the number of cells are sufficient to produce the amount of factor VIII that is required with them. Sometimes, instead of half maternal X getting inactivated, half maternal X getting inactivated, which is normal, sometimes in a great proportion of cells, the same parental X gets inactivated. That means out of 5,000 cells, something like, say, 4,500 cells, paternal X has got inactivated. That means this is not random. Such type of inactivation is called as skewed X inactivation. Skewed X. Skewing of the inactivation. Why does this happen? Let's start off with simple things first. In one of the X, either maternal or maternal, the XIC has mutated. In such a case, that X cannot get inactivated because there is mutation of XIC itself. Automatically, the other chromosome which has got normal XIC will get inactivated in much more than 50% cells. Okay? That's one reason. Second reason, if a segment of X has been deleted, but not the XIC, the segment of X is deleted, visible loss, not point mutation, visible loss, structural deletion, it will be prudent that that X should get inactivated because there are lost a lot of segments and a lot of genes will be lost. Luckily, that's what exactly what happens. When there is a structural abnormality to one X chromosome, but with the presence of XIC intact, that X will get inactivated in much more than 50% of the cells. So structurally defective X gets inactivated in the presence of XIC. In the absence of XIC, normal X gets inactivated. Is it clear? Second point, second reason. Suppose there is a structural defect, and this structural defect is translocation. A segment of X has gone to an autosome, a segment of autosome has come to the X chromosome. Exchange of segments. That X, which has got a translocated autosomal piece in it, will not get inactivated because fully functional both autosomes are required. So you can't have a piece of autosome getting inactivated. In this case, normal X gets inactivated. Are you getting the point? Fully functional, all autosomes are required for activities of the body. So we saw three conditions where there are skewed X inactivation, when the XIC gets mutated or deleted, normal X gets inactivated. When there is a structural abnormality of X chromosome, but with the presence of XIC, defective X will become inactive. And if there is a translocation of an autosomal segment to the X chromosome, you cannot have that X getting inactive because then that autosomal segment will also get inactive, which is not permissible. The normal X gets inactive. This is the reason why certain individuals, females, who have one X defective for hemophilic factor and second X is normal, we said they produce normal amount of factor 8. But if there is skewed inactivation in such a manner, the X containing normal hemophilic factor has become inactive in much more than random inactivation, then the only functional X is the abnormal X having a hemophilic factor. This condition is called as manifesting heterozygote in female with X-linked recessive disorders. Manifesting heterozygous. Heterozygous means one normal, one abnormal. They are called heterozygous. So normally in the recessive condition, heterozygous should not manifest in females. But because of the skewed X inactivation, which is also called as total lionization. Skewed X inactivation, also called total lionization, where predominantly almost all one parental X has got inactivated. This can produce a condition called as manifesting heterozygous in female recessive disorders. Remember, don't use the word recessive gene, dominant gene. It's a condition which is recessive or dominant. After that we discussed that ribose sugar, which is the formula of C5H10O5, which is a pentose sugar. When second carbon loses one oxygen molecule, second carbon losing one oxygen molecule produces C5H10O4. This is called as deoxyribose. So which carbon loses oxygen? Carbon with second number. First carbon has got an attachment of a nitrogen base. A pentose sugar with the nitrogen base is called as nucleoside. The nitrogen base can be purine, that is adenine or guanine, or it can be pyrimidine, cytosine or thymine, or methylated thymine, which are called uracil. Nucleoside containing adenine is called as adenosine. Guanine is called guanosine. Cytosine is called cytidine. Thymine is called thymidine. And uracil is called uridine. When the fifth carbon gets a phosphate radical, it is called as nucleotide. If there is only one phosphate radical, it's called monophosphate. Two phosphate radical, diphosphate, and three called as triphosphate. And we have all heard of the celebrated ATP, adenosine triphosphate, which after the release of energy, it becomes diphosphate, till it can be triphosphated again.
Phosphorylation.
If there is one phosphate at the 5' end of a molecule gets attached to another nucleotide, it will get attached to the third carbon of the other nucleotide. This is called as dinucleotide. Well, there are two of them. The fifth carbon of one is attached to the third carbon of the other. So now we have two nucleotides, where one is bound by 5'3", the other nucleotide, the front nucleotide, has got the 5 which is having a phosphate which is free. That end of the nucleotide is called as 5' end. The other end is called 3' end. Because the 3' is a potential site of attachment of another phosphate there. When you have a series of nucleotides, it's called as polynucleotide. We also discussed that purines, adenine, guanine, catabolism produces uric acid. Excess of uric acid produces a clinical condition called gout. Another thing about the union is that adenine has an affinity to thymine, purine pyrimidine affinity, and guanine has an affinity to cytosine. When these come together, they are held to each other by hydrogen bonds. It was found that when you heat the DNA, the hydrogen bond breaks. The energy required to break the hydrogen bond between cytosine and guanine is 1.5 times the energy required to break the hydrogen bond between adenine and thymine. Therefore, we say there is a double hydrogen bond between A and T, and there is a triple hydrogen bond between C and G, because that's why 1.5 times. So, when one nucleotide, polynucleotide, has got one 5' end, the other 3' end, another polynucleotide will form a binding with this nucleotide in such a manner that the nitrogen base of A will pair with T, T will pair with A, and G will pair with C, and C will pair with G. So, one of the polynucleotide has got one 5' and one 3' end. The other nucleotide has got a reverse 5' end and 3' end, and the nitrogen base is pairing as per AT and GC, becomes a double nucleotide parallel to each other, but since the direction is reverse, we call it as these two are anti-parallel to each other. They are parallel, but they are running in different directions. So anti-parallel to each other. The pentose sugar here will also be deoxyribose. This is what is called the magic molecule, the miracle molecule called DNA. This is the only known molecule which has got the capacity of spontaneous replication. Okay? Why only this has, why nothing else has, we have no idea absolutely. But this is capable of self-replication. This is also capable of transcribing to form an mRNA, which can be translated to form a polypeptide, which can get folded in different shapes to become a protein. So the segments of DNA which are responsible for protein production are called genes. Segments of DNA which are capable of protein synthesis are called genes. There are 3 into 10 raised to 9 nucleotides in a haploid set of chromosomes. 3 into 10 raised to 9, which, considering the completely unfolded DNA, will come out to be somewhere around 2 meters or so in a single cell. If you open up the DNA, 2 meters long, haploid set. Now, how much percentage of this DNA will be meant for protein synthesis and how many do not produce protein synthesis? Approximate ratio. How many percentage of this DNA are genes and how many are non-genes? It is said 65% of the chromosome DNA are genes. But when you say gene, the whole gene does not get transcribed because there is within that called as a regulator gene. Within that segment there is a promoter gene. Within that segment is an operator gene. Within that segment is a transcribing gene. Within the transcribing gene, there are segments which are going to be removed later, called as exons, called introns, and those which are actually going to be transcribed are called exons. Introns are interspersed within the coding segment, which has to be removed before it becomes an mRNA, functional mRNA. So finally, if you look at it, only 2% of the entire DNA sequence are actually responsible for coding of proteins. That's why you heard the discrepancy. 65% of DNA are genes, 35% are intergenic. But only 2% of the genes actually code for a polypeptide. The remaining are all those, like typical Indian family, when they are going far off, 65 people will come into the railway station to see the 2 people going. Okay? That's why tickets may be minimum 5 rupees, platform ticket is 10 rupees. Because they earn more from the platform tickets. 35% which are intergenic, they are repetitive sequence. The repetitive sequence can be interspersed repeats scattered throughout the genome, or they can be tandem repeats one after the other. Interspersed repeats are 25%, and they are long and short. Interspersed repeats are 25%, and they are long and short, and called as LINE and SINE, lines and signs. Where line stands for long interspersed nucleus element. No prize for guessing that sign stands for short interspersed nuclear element. So the name suggests some are long, some are short, but they are scattered, they are interspersed throughout the genome. 10% of the repeat sequence, we said 65% are genes, 35% are intergenic, which are interspersed or tandem. 10% which are tandem repeats are repeats of anywhere from 2 to 10 sequence of nucleotides. Repeated one after the other. And these tandem repeats, how much should be the number of tandem repeats is dependent on our parents. Two individuals except identical twins will not have the same number of tandem repeats. That's why it's called as VNTR, variable number of tandem repeats. VNTR is what is used for DNA fingerprinting. So what to do? Identify the tandem repeats, the sequence, and break that down. Put it on an electrophoretic plate. Larger number of tandem repeats together will move slowly along the electrophoretic gel. Shorter ones will move faster, producing different bands. Match it with the parental bands. Each band of the child will match with one of the two parents. This can be easily used in cases of disputed paternity. The child will have half the gene, half the bands patterning with mother, and the other half does not pattern with the putative father, disputed paternity. Very extremely unlikely for the person who is not the parent, except as we said, identical twin. It will not have a perfect match to all the bands which are produced from the child. Is it clear? That's the basis of DNA fingerprinting. But the point to be noted is actual physical fingerprints or even two identical twins are different. DNA fingerprinting is not. Why they have similarities in their fingerprints, but not identical. What causes the fingerprints to form different, even in identical twins, is completely unknown. It has to do something with the cosmic manifestation in the fingerprint. Probably because the person was on the left side of the uterus and other was on the right side, different cosmic rays, different fingerprints. So fingerprints are defined by genes, but not exactly only genes alone. Okay? The remaining 65%, which are coding for genes, has been counted on a haploid set because they've already done the complete human genome analysis. HUGO project. HU, human, GO, genome. Genome refers to haploid set. So, Human Genome Project or HUGO project was completed in 2003. Before that, we know that the number of polypeptide proteins which are produced by the body are 1 lakh in number, all the proteins. We felt there are 1 lakh genes, one gene, one protein. That was what the former belief. It was found that 80 rich regions are poor genes, have fewer genes than GC rich regions. Segments of DNA which are GC rich are likely to be the genic part, gene rich areas. Segments which are 80, more 80s are gene poor region. That is the reason why whenever there is a break in a chromosome during translation, inversion, translocation or whatever, the breaks usually occurs in the gene poor region. Because 80 band is easier to break than the GC band because of double hydrogen bond and triple hydrogen bond. That is the advantage is the fact that the gene poor region are broken. That gene where the break occurs, that area where the break occurs, that may not have any gene. So if the gene, when the chromosome is inverted in its place or translocated to another place, the gene does not break most of the times. And rarely it may break through the genic region, where it can result in varieties of cancers. So the first cancer which was described was people with chronic myeloid leukemia had a reciprocal translocation between chromosome 9 and 22. This was first described at a scientific conference in Philadelphia. That small chromosome 22 was called as Philadelphia chromosome. It had exchanged a big segment to 9 and received a small segment from 9. So it became even smaller than what it was. Are you getting the point? Similarly, 8 and 14 translocation through a strategic point can produce Burkitt's lymphoma. But these are very rare. Most of the times the breaks do occur, but they occur in the intergenic anti-rich region, which are gene poor. So based on the gene-rich region, they calculated that the number of genes should be 1 lakh, because there were 1 lakh proteins. After the Human Genome Project was done, they found out that there are only around 20,000 genes. That means 5 times lesser than what was earlier expected. If there are 20,000 genes, how are 1 lakh proteins produced? After the DNA is transcribed to mRNA and mRNA is translated to become proteins, there are some modifications occur before these things. It's called as post-transcriptional modification and post-translational modification. So based on these, one gene can produce more than one type of protein, depending on the modification. One of the modifications is just an intron removal. In some cases, some introns may not be removed if you want to form a different protein. In some cases, they will be removed. So different proteins, different mRNA, different. Secondly, even after the polypeptide is formed, different styles of folding will make different proteins. Okay? And pairing up of proteins, quaternary rearrangements. There's primary rearrangement, which is folding of polypeptide. There's secondary rearrangement, tertiary rearrangement, different patterns of folding of proteins, and pairing up of multiple proteins in different format produce a different functional unit. This is called as post-translational modification. So, on an average, one gene produces approximately 5 proteins. It's purely average. Some may produce more than 5, some may produce less than 5. For example, dystrophin gene produces only dystrophin protein, nothing else. So there are some one gene, one protein, but one gene can produce otherwise multiple proteins. These genes are also defined as unique sequence of polynucleotides because others were repeats. This is a unique sequence. That means one gene on one haploid site will produce a specific type of protein. Only that region will have similar site. There will be no repeats anywhere in the elsewhere in the DNA. Say for example, one segment produces beta globin of hemoglobin. Only that site will produce beta globin. No other sites in the haploid genome will produce the beta globin. Of course, if the homologous pair received from the other parent, you will have. One prominent exception to this is alpha globin of hemoglobin. Remember, adult hemoglobin are alpha to beta 2. Two alpha globin, two beta globin. Variant of adult is alpha to delta 2. Fetal hemoglobin are alpha to gamma 2. Embryonic hemoglobin alpha to epsilon 2. So alpha is required for every type of hemoglobin. So nature in its wisdom gave two alpha genes close to each other on the same chromosome, but two, then in case one gets damaged or lost, at least we have the other one. So every individual normally has 4 alpha genes in every somatic cell, 2 received from each parent. They are close by to each other. During pairing up of meiosis, alpha will pair with alpha. The other alpha will pair with the other alpha. Both are quite close to each other. But sometimes one may pair with the upper alpha will pair with the lower alpha because they are homologous. And then the crossing over and twisting occurs. You will have variable number of alpha in the child. So it is possible that one of the individuals may have 2 alpha received from one parent and only 1 alpha received from the other parent because of the mismatch crossing over. 3 alpha are still there, no problem. This type of mutation is called silent mutation. You did not even realize that one of the alpha is missing. Now during meiosis of that individual, it is possible that another alpha will get lost. And a child will receive no alpha from one parent and two alpha from the other parent. Now this person will show symptoms of alpha thalassemia, but not significant enough. Two are still there. This is called as thalassemic trait. If the thing continues further, three alphas are lost and only alpha is being overloaded with production. You get alpha thalassemia minor, which is also called as thalassemia. If all the four alphas are lost, the child will not survive. That's called hydroxythalis. The thalassemia minor and major are usually restricted to beta, because beta there are only two copies. One lost thalassemia minor, two lost thalassemia major. But here there are four variants. Normal, 4 alphas. Silent, 3 alphas. Small degree of trait, 2 alphas. Severe thalassemia, 1 alpha. Hydroxythalis, low survival, low alpha. Okay? So even though we use the word unique sequence, some repeats are possible in some such cases. So we have two genes producing the same protein, but 20,000 genes, 1 lakh proteins. Tandem repeats one are also called as satellite DNA. Satellite. Why? Because we pair up with the other segments of DNA, they are short, very short tandem repeats. So if you break down the repeats, they become very small molecule, 2 to 10 nucleotide. And they will randomly pass through the electrophoretic gel and be very far away from the other bigger molecules. They appear like the bigger molecules are the Earth, and this is shot away from the Earth, satellites. Don't confuse satellite DNA with satellite chromosomes. Okay? To understand satellite chromosomes, you need to know karyotyping. Okay. Which cell will you choose for doing karyotyping? I'll give you 4 options. Epithelial, connective, muscle or nerve?
It means epithelial?
Epithelial, connective, muscle or nerve? See, muscle nerve are useless because they don't multiply. You want the cells to multiply. Epithelial and connective are 2 possible options. Epithelial cells are attached to each other by desmosomes. So they don't separate out. You want to study individual cells. Group of cells cannot be studied. I'm getting the point. So we use connective tissues. For bar body, we can use epithelium. Because you're studying individual cells, individual nucleus under light magnification. Here you require isolated cells, therefore connective tissue. And connective tissue cells, what will you use? Blood or tissues? You can use fibroblasts from dermis, or you can use lymphocytes from blood. Which is easier? Lymphocytes from blood is easier than fibroblasts from nervous. Nervous has got so many different varieties. It won't become fibrosis, it becomes something else. But blood has got thousands of RBCs and only few lymphocytes. How will you isolate the lymphocytes? In a heparinized blood, you centrifuge it. Upper 45% is plasma, lower 55% is RBCs. In between the two, there are 2% of white blood cells. It's called the Buffy coat. From that you isolate the lymphocytes. Okay? The other two alternatives which are used for karyotyping is amniogenic cells. So remove the amniotic fluid, centrifuge it, cells will lie at the bottom, they are called amniogenic cells. The fluid can be used for biochemical tests, but cells can be used for karyotyping, amniogenic cells. The fourth option is chorion villus cells. Chorion villus are not picking up embryonic cells, you are picking up from the placenta, the chorion, even before the placenta is formed. So around 10th or 11th week of intrauterine life, you can pick up the chorionic cells. It requires around 14 to 18 weeks to pick up the amniotic amniogenic cells because uterus becomes abdominal only after 12 weeks. So what are the four cells we use for karyotyping? Fibroblasts, lymphocyte, amniogenic cells and chorion villus cells. We will focus on lymphocytes. Lymphocytes are placed in a culture medium. And you put a mitotic agent in it. So it repeatedly undergoes mitosis. The other white blood cells do not undergo spontaneous mitosis like lymphocytes do. So that's why we chose the lymphocytes. Cells keep on multiply, then you put after 3 days of sufficient multiplication, you add colchicine. What is colchicine? Treatment of gout? Colchicine? Colchicine is treatment of cancers. It's an anti-cancer drug.
Inflammatory gout में use करते हैं.
अरे? Bad. Anyway.
नहीं, not anymore, sir. पहले.
They didn't know what it was causing. It goes and kills those cells. Anyway, so what happens is, colchicine acts by preventing the spindle formation. If spindles are not formed, chromosomes will not separate. If chromosomes don't separate, the cells will get arrested in metaphase part of cell division. Metaphase, the cells are like the equator and they are waiting for the spindles to separate them. Anaphase will not proceed because of the absence of spindle formation by the colchicine. That's exactly how it acts in cancers also. Do not allow the cells to multiply. But do not allow normal multiplying cells also to multiply. It's like hair follicle. That's why loss of hair. Ulcers, skin, GI. All those are aplastic anemia. These are all the side effects of anti-cancer therapy. Coming back to, after that, you introduce hypotonic saline. What will hypotonic saline do to the cells?
Water will dehydration. Hypotonic muscle.
Hypotonic saline.
Swelling of the cells.
It causes swelling of the cells. Water will enter the cell to try to balance the tonicity. Cells will swell, so it will rupture easily. Drop the cell from a height onto a clean glass slide and hope that only one cell breaks. So you have to do it repeatedly on multiple slides and hope many of them when only one cell breaking. Otherwise, you will count the chromosomes and say, hey, this is 92 chromosomes. But actually have two different cells mixed up with each other. So repeatedly you do it, and with practice you know what height, how to pick up less cells at a time, or maybe one cell at a time, drop it to a height, it will burst. If it bursts, the chromosomes which are already arrested and the nuclear envelope is gone, the chromosomes will get dispersed. And hope that they get dispersed, all of them separate, not that one or two of them overlap each other. So you require practice to drop for a specific height, they get properly dispersed, and not so much that they fall out of the slide. All those things require great expertise. But then once the cells are fully, chromosomes are fully scattered, then you use, if you directly use GIMSA, it will stain all cells are black. That was what was done for the first time in Denver 1965. They used GIMSA and stained all chromosomes as black. So at that time, you could classify chromosomes tall, medium size, short. And tall also, metacentric, submetacentric. Okay. And nowadays what we do is, we put trypsin. Trypsin denatures GC-rich region. Trypsin denatures GC-rich region. That means the genes. It does not denature AT-rich region. Now poor GIMSA. The chromosome which has portions of chromosome which has not been denatured will stain with as black. And those which are genic regions, gene-rich regions, they do not stain. So you can alternate dark and light bands.
So gene-rich region will become
Black. The gene-rich regions are denatured, na? So they will remain uncolored.
Okay.
And gene-poor region, AT-rich region, which are not denatured, will become black. So the uncolored portion are the genes, and the colored portion are the gene-poor. That's what is done for banding. If instead of using trypsin, if you heat the cell slightly, you will find that the AT region will break first, then the GC. So heat to such an extent that only the AT will break, and then stain. you will find that the AT region will become uncolored and GC rich will become colored. So the first type of banding was called as G banding. This also uses GIMSA, but it produces a banding which is a reverse of what was there earlier. It's called R banding. Most of the time they use G banding because it's very easy to measure how much trypsin is to be used rather than heat to exactly such an extent that AT breaks the GC result. Based on the banding pattern, every chromosome could be described. And its centromere was also easily identified. So morphology of the chromosome was also appropriate. Autosomes are numbered based on the descending height. This was numbered in 6 years after Denver classification. This is called as Paris classification. Paris. 1 to 22. And the sex chromosomes are called as X and Y. And we said there is decreasing order of the heights. Okay? This was done in 1971. Based on this classification, which chromosome do you think is the smallest of all chromosomes? 22. That's what we expect it to be. With the modern exact measurement of the nucleotide sequence, etc., it is found that the smallest chromosome of all is chromosome 21. They made a mistake in 71. The microscopes were not all that good. And they found out that chromosome 21, which they called then as larger than 22, that's why they named it 21, show 3 copies in individuals with Down syndrome. So Down syndrome had 21 trisomy. And this was the first chromosomal disorder which was confirmed that is a chromosomal excess. All others came later. Although Dowd had described this in 19th century, the confirmation came only after 1971. Because numbering of chromosomes came on. By then, 21 trisomy Downs was so much ingrained and established in people's mind, including scientists, they found it difficult to swap the change of numbers. So Elphinstone Road can be called Prabhadevi. 21 could have been called as 22. But Down syndrome is 21. They have stopped, they have given up trying to rename chromosomes. You identify chromosome 21 not by the length, but by the bending pattern. Small deletion of one chromosome may result in alteration of their height. Bending pattern is unique. Totally in a haploid state, how many bands do you think will be there? Approximately 400. Okay? Now they have got techniques to arrest the chromosomes in prophase, not metaphase. Metaphase where the chromosome is maximally condensed. Prophase or prometaphase, just before metaphase, the chromosome is more slender. More slender means more bands. High resolution banding techniques will show show you around 950 or more bands. So what was 400 bands in Android set became 900 plus in a high resolution bending. Small deletions segments can also be picked up, which is a metaphase bending it could not. Okay? So high resolution bending for identifying small segments, loss or doubling. What is the role of folic acid in cell division? Important. It's important. Which is why pregnant women, even before they know that they are pregnant, are given folic acids. It has been shown to prove that it produces lesser neural tube defects. Because the brain is a cell which grows very fast. And for that fast cell division, you require more folic acid. You know, in Britain, if you go to your family physician and say that we have decided to have a child, the physician will give them folic acid. Because even before you know you're pregnant, you get to know that you're pregnant 3 weeks after fertilization. By then, brain growth has already started. And folic acid is less, neural tube defects. So start administering folic acid right then and there. And because it's a water soluble, there's no such thing as excess folic acid. America goes one step further. There they don't know that they're going to get pregnant. So they have fortified their bread with folic acid.
Like we have iodized our salt.
Iodized salt, they have folic acid enriched breads. So that even without knowing you are consuming enough folic acid. So if the cells, while dividing, they are kept in folic acid deficient medium, the chromosome will divide, but there will be some errors. In such a case, what has been found out is that trinucleotide repeats are the region where the effect is maximum. So they found out that trinucleotide repeat sequence diseases like fragile X syndrome. The fragile site is seen only in folic acid deficient medium you are doing karyotyping. You have not seen ordinary karyotyping or even higher resolution. It's a CGG trinucleotide repeats which has occurred more than 200 times tandem. More than 200 tandems, there will be picked as fragile. Less than 200 times tandem, it will not be picked as fragile. So, the number of repeats are usually 10 to 20. When it becomes 100 to 200, it's called pre-mutant. When it exceeds 200, it's called mutant. 200 to 500, mild mental retardation. 500 to 1000, moderate. Over 1000 repeats, severe mental retardation. Fragile X syndrome is the commonest cause of male mental retardation. It will occur only in males because there is other X in males. Okay? So fragile sites cannot be picked up even by high resolution binding. Then there are bindings which will pick up only the C region. C C region is centromeric region. Centromeric region is a repetitive sequence. There are no genes very close to the centromere. If you stay only in the centromeric region, that's called as C banding, you can easily count the number of chromosomes. You are not bothered if the chromosomes partially overlap or not. You can count how many black dots are there. Nowadays, they have got C fluorescent labels. C region is not coding for proteins, but the C region of every chromosome is unique. C label of Y chromosome is what we discussed earlier and what is used in Olympics to identify how many Y chromosomes are there. They are using Y chromosome C sequence labels added with fluorescent dyes. So you actually study C regions of all chromosomes. If you put 21 labeled fluorescent dye in a person with classical Down syndrome, you will find 3 fluorescent dots. And this is used in interface. So all those elaborate karyotyping is not required. 3 fluorescent dots of 21 label means 21 trisomy. Yeah? They have gone one step further. C is unique for every chromosome. Other segments are also unique for different chromosomes. So we put multiple labels, multiple fluorescent parts, but make sure that one chromosome label has got a fluorescent of one color. All the labels of one chromosome having one color, another chromosome having different color, and 20 different colors. Fluorescent tag, if you give it. It will paint the whole chromosome. It's called as whole chromosomal painting. Now, if there is even a very small translocation, you can pick up where this color has joined that particular chromosome. You will say, what are the 21 colors? And they're all fluorescent, huh? So fluorescent green, fluorescent light green, light red, blue. How many colors can you identify in fluorescent dyes? You don't bother, computer will do that. And it will become even slightly different shades of color, which we will not be able to do. But whole chromosomal painting looks so nice. And every chromosome is painted with a different shade. Right. In this karyotyping, what was found that chromosome number 13, 14, 15, 21 and 22 only. 13, 14, 15, 21, 22. All these 5 are acrocentric chromosomes. What is acrocentric?
The centromere is in the
near to one pole.
Then what is submetacentric?
Only one acrocentric, one center.
Let me go into the 4 types of chromosomes. If the centromere of a chromosome is close to the center, not exactly in the center, close to the center, it's called as metacentric. That's why the word meta. It is not centric chromosome, metacenter, next to the center. If it is close to one end, not exactly at one end, close to one end, it's called an acrocentric chromosome. Acro means tip, T-I-P, tip, like acromion, tip of shoulder. If it is neither close to one end nor close to the center, somewhere randomly in between, it is called a submetacentric. If it is bang at one end, bang at one end, the other arm is not there at all. It's called as telocentric. Now, how close is metacentric to the center? When you call it metacentric and submetacentric? So there is a ratio. From 1:1, short arm, long arm versus short arm. If it is 1:1, up to 1:1.7. From ratio of 1:1 to 1:1.7 is sufficient enough to be called as metacentric. Okay? If it is between 1:1.7 to 1:3, it's called as submetacentric. Whereas anywhere from 1:3 to 1:7, acrocentric. Telocentric, there are no examples in humans.
So in humans only three types of
Only the types. And you know, in the Denver classification, we have classified it as A group, now called chromosome 1, 2, 3. They are tall metacentrics. B group, tall submetacentrics. C, D, E group, medium size. Submetacentric, acrocentric, submetacentric. The F, G are the short chromosomes. E, F, G actually are short chromosomes. Anyway, all these things is now obsolete simply because the celebrated chromosome 1, 2, 3 has a tall metacentric. 2 has been found is actually submetacentric. All those theories which they had, tall metacentric, tall, is all gone. So we now call chromosomes as 1, 2, 3, 4 up to 22, X and Y. Interesting are these 5 chromosomes which I am talking about. 13, 14, 15, 21, 22 are all acrocentric chromosomes. Y is also an acrocentric chromosome. Y. X is a submetacentric chromosome. Forget Y. Only these 5 autosomes, 13, 14, 15, 21, 22, of one parent and the other parent. These acrocentric chromosomes are specialized. They have got a centromere, they have got a long arm. The short arm is where the interest arises. The short arm has got an extremely small part attached to a centromere, and a large part of the short arm, which is also very short, is away from centromere, and in between the two there is a gap. So this was considered as satellite. The fact that they were called satellites is that wherever you pull the chromosome, the satellite moves with it. Like wherever the earth revolves, continues to revolve around the earth one. That's why they are called satellites. Further improvement in microscope found out that the satellites are not true satellites. The two short segments of the short arm were connected to each other by a narrow constricted portion, which is called a secondary constriction. Centromere is the primary constriction. And the secondary constriction, which connects the satellite with the rest of the chromosome. That's why obviously, if you move the chromosome, the satellite will move with it. It's not satellite. Something like connecting Earth to Moon. Move the Earth, move will move with it. But more interesting than this phenomenon is that portion of the satellite is responsible for production of rRNA, ribosomal RNA, which is from ribosomes. Now, what do ribosomes are responsible for? Protein synthesis. So they are abundantly placed in cytoplasm, especially those which require to form larger amount of protein. And these rRNA are produced by these chromosomes. So it will first form ribosomal RNA, then it will get transcription always occurs in nucleus. Translation always occurs in cytoplasm. Even proteins which are required in the nucleus, they will translate in the cytoplasm and enter through the nuclear pore. So when ribosome requirements in the body are large, especially highly metabolically active cell, ribosome RNA production will also be large. Under microscope, with good resolution, they found in certain cells a nucleus and some halo region within the nucleus. They were called as nucleolus. Nucleolus is a site for rRNA production. How many nucleolus lies in a cell? I'll give you options. 0, 1, 2, 3, 4. How many nucleoli lie in one nucleus? 0, 1, 2, 3, 4. All 5 are correct answers. Depending on how active the cell is. If the cell is extremely inert, it will not show nucleus. It will not show nucleus. Not that it does not have nucleus. Lesser amount. So you don't see the nucleus. The most active cell can show up to 4 nuclei. But this is in one plane. Potentially, there are as many as 10 nuclei that can be present in a cell. 13, 14, 15, 21, 22 maternal, 13, 14, 15, 21, 22 maternal. Y chromosome is also acrocytic, but no satellites. Therefore, no rRNA production. But usually, as you said, 4 nucleoli are noticed there in highly active cells. Others are not active now. Obviously, it goes without saying, if out of those 10 nucleoli producing region, they are called as stated by silver dyes, and they are called as AGNOR staining. AGNOR. Silver AG, nuclear organizing region. People think it's a misprint. They think it's a silver nitrate, AgNO3. It's not the it's R, AgNOR, nuclear organizing region. And what was I saying? That silver, there are 10 possible nuclei. If out of these 10, 2 are lost, what will be the effect? No effect. Because after all, 8 are still there. And even the most active cell do not have more than 4 nuclei at a time. So, whole segment of chromosome is gone. Still no effect. Only this, because there are 10 potential sites for producing RNA. Robertsonian translocation is a special type of translocation which occurs only in acrocentric chromosome, where the 2 long arms fuse with each other and both the short arms of the chromosome are lost. 2 long acrocentric chromosomes fuse with each other. So you get a new chromosome which has got long arm of one chromosome and long arm of another chromosome. And both the short arms are lost. No problems at all for that individual because 8 of them are still there. Problems will occur during meiosis as homologous segments try to pair up with each other and they are searching, where is my pair? So errors in meiosis is almost 30 to 40%. So the history will be a repeated spontaneous abortion, 30 to 40% of the times. Okay? So recurrence risk is that high. Anything more than 10% is considered high risk. Coming back to acrocentric chromosomes, interesting one. Last comment about telomeres. What are telomeres?
The centromere is at one end, and one of the arms is
Ends of chromosomes are called telomeres. So there is a P telomere
telomere.
and Q telomere, ends of every chromosome. Telomeric region are also non-coding. So the telomeres are lost. There is no problem. The chromosome will encircle and form a ring chromosome. Both the telomeric deletion will cause a ring chromosome formation because the cut ends are called as sticky ends. And the 2 sticky ends will fuse with each other. And telomere being non-coding, nothing much is lost. Unless you have got a big segment of ends are lost, along with that few chromosomes, genes are also lost, then there will be a problem. Otherwise, the ring chromosomes should not have a problem with that individual. Problems will occur in meiosis because the pairing becomes a problem. We said telomeres are non-coding, which means it's a repetitive sequence. A specific code repeat of all telomeres are 50 or more in a zygote. With each cell division, the repeat telomeric sequence reduces. With each cell division, the number of repeats reduces. After 50 cell divisions, the cell cannot divide. Cell cannot divide. And the cell is said to be senescent or old. Dolly the sheep was produced by taking a mammary cell of the mother and putting in a new fertilized nucleus within it to form the new clone. But remember, that cell which was there in the mother, if it's compared to humans, were already divided 25 times, approximately. So now, there is only a 25% times that cell can divide, and that cell will become old. Sheep become old by the age of around 14. Dolly showed signs of old age at the age of 7. Understandable, because you have taken the cell which was already divided half the times, whatever the number for sheep. And they say 25 because we are comparing with humans. So ideally, if you want to make a clone, which has got near normal survival, you have to take embryonic cell, which is divided least number of times. And nowadays, they have a bank where they keep umbilical cord cells in cold storage till the age of around 21 or so. Charge a hefty sum of money, but whenever the organs are required, you can always get it in the remote possibility that you may require it. But at least there is some hope that this is a. But who can afford that 21 years cold storage? And what are the chances you will require anything? What are the chances that stored one will actually help you? But when you have money to burn, you can always do such thing. Anyway, coming back to, so that's the thing. During meiosis, telomeres are restored back to the original number. So meiosis are not just a special type of cell division as compared to mitosis, they are very special. They are just special. Reduction division, two cells, two cycles, equational division. That's all passe. Three extraordinary things which occur in meiosis. One, exchange of segments. Such a thing is unheard of in mitosis. Exchange of segments. Second, restoration of telomeres. So the potential embryo has got chance of another 50 cell division. And third, what is even more mystical, Americans produced a wire and showed it to the world. World's thinnest wire. World's thinnest wire. Thinnest. Japan took that wire, using their nanotechnology, they buried a tunnel through and through the wire. Thinnest. The Indians took it, show me, and returned it back. They looked at the wire, looks similar. What have you done? Said, read inside the tunnel. Is written as made in India. Anyway, this is just a joke. The reality is, every chromosome that we have, 50% is derived from father, 50% is derived from mother. They are called paternal and maternal. And you can identify which chromosome is maternal set and which chromosome is maternal set. Okay? This phenomenon is called genomic imprinting. Every genome, every chromosome has got parent of origin label. During gametogenesis, this label is wiped out. During meiosis, this label is wiped out and a new label comes to those cells, all cells. If you are a male, it will come as paternal label. If it's a oogenesis, the label will be maternal. Because you are going to be the mother or father of the next generation. So that the child will get paternal label from the father, even if the cell chromosome actually means father's mother. The father had that chromosome labeled as his mother. But when he transmits the chromosome to the child, it will have maternal set. That means the labels are wiped out during meiosis and a new label comes. So these are the three extraordinary things which occur in mitosis. Most people know only exchange of segments. But restoration of telomeres, and that's why when we said cell can multiply only 50 times, certain viruses produce telomerase enzyme. That will cause further multiplication. So cancer cells which keep dividing without stopping at 50 generations must be having telomerase enzyme, which is allowing them later division. Genomic imprinting is important because chromosome 15, the long arm of chromosome 15, remember 15 chromosomes is an acrocycline. Short arm we have already discussed. The long arm of chromosome 15 has got a segment whose deletion produces Angelman syndrome. The same segment deletion sometimes produces Prader-Willi syndrome. Prader-Willi. How can the same deletion sometimes lead to Prader-Willi and sometimes to a completely different Angelman? It was found that if the chromosome 15, that Q-arm deletion, segment of Q-arm deletion, belongs to the paternal set chromosome 15, it produces Prader-Willi. If the deletion is of the maternal set, it will produce Angelman, genomic imprinting. Okay? That means even though we say they are homologous chromosomes, they still have different functions. All 46 cells are responsible for cell division, but the control of cell division is by one set. Trophoblastic set control is done by paternal set. Embryoblastic proliferation is controlled by maternal set. So all those experiments where we do seedlessness, what is called as parthenogenesis and parthenocarpy, will not work because mothers will have two maternal sets. Trophoblasts will not run. It's a very chauvinistic belief, but that's a fact. That father brings the food, mother looks after the baby. What was the side effect of this is that suppose there is dispermy. One ovum has been simultaneously fertilized by two sperms. Net result 69 chromosomes. 23 into 3. Twenty-one trisomy Down syndrome. Smallest chromosome, one extra copy, Down. 18 chromosome, Edward syndrome, one slightly longer chromosome, more side effects than Down. 13 trisomy, Matau syndrome, more severe defects. If Matau can survive few minutes to hours, Edward can survive hours to days. Downs can survive days to months, sometimes a few years. Whatever it is, what we are trying to say is that one extra chromosome, if produces such a large effect, what do you think? 23 extra chromosomes will result in spontaneous abortion. But before the spontaneous abortion occurs, trophoblast will grow faster than embryoblast. This condition is called as vesicular mole. This 69 chromosome has no hope for survival at all. Nature in its wisdom, if that individual is meant to survive, then one of the set of chromosomes has to be spontaneously destroyed. If one of the set of the chromosome is spontaneously destroyed, then he has any hope of survival. Mathematics says three sets of chromosomes, 23, 23, 23, 69. Two paternal set, one maternal set. If you spontaneously destroy one, there's a one-third chance that the maternal set will be destroyed. What will happen? No growth of embryo. And no control by the mother. If the cat is away, the mice will play. The trophoblast proliferate at such a rapid pace, it results in choriocarcinoma. Where it was benign in 69, it becomes malignant in 46 with mother cell destroyed. If one of the maternal cell was destroyed, normal child. Which means genomic imprinting does come into play even at lactocystic stage. That means it was present at the zygote level itself, which was the new imprint occurred during meiosis. Okay? Can we stop for today?